So beyond the tubes themselves, what happens once the sample is collected? How do they get it from, say, the patient's arm to the analyzer without degrading?
Right. If we connect this to the bigger picture, general handling, transport, and storage are equally vital parts of that pre-analytical phase. Many samples, as we touched on, have what we call time sensitivity. They require immediate analysis or transport, often under specific conditions, to prevent degradation.
Like the arterial blood gases you mentioned.
Exactly. Blood gases, ammonia, lactate, certain coagulation tests, CSF samples for cell counts. These often need to be analyzed almost instantly or at least processed very quickly. Delay can significantly alter the results.
And temperature? Does that matter much?
Oh, hugely. Temperature control is paramount. Many urine specimens, especially for 24-hour collections or if analysis is delayed, need to be refrigerated. Same for many serum or plasma samples after they've been separated from the cells. Stool samples for C. difficile toxin testing or occult blood often require refrigeration too. CSF might be refrigerated if cell counts aren't done immediately, though room temp is preferred for cultures.
But you also mentioned some things must never be refrigerated.
Correct. This is where it gets tricky, and knowledge is key.
Mm.
Certain tests are for analytes that are cold sensitive. Cold agglutinins, for instance, are antibodies that cause red cells to clump in the thyroid, so that sample must be kept warm, usually at body temperature, 37 degrees C, from collection through testing. Cryoglobulins are proteins that precipitate or gel in the cold, so again, keep warm. Anaerobic cultures, which look for bacteria that grow without oxygen, can be killed by cold temperatures, so those stay at room temp. Routine stool cultures often stay at room temp too, as refrigeration can kill off sensitive bacteria like Shigella. And platelets for transfusion need to be kept at room temperature with constant, gentle agitation to remain viable. So hot, cold, room temp, it all depends on the specific test.
Fascinating. What about light?
Ah, yes. Light protection is crucial for certain light-sensitive analytes. These are substances like bilirubin, especially important in newborns, urobilinogen, porphyrins, and some vitamins like folate and vitamin A.
Why light sensitive?
Because exposure to light, especially UV light or even strong fluorescent light, causes these molecules to break down to degrade. If that happens before analysis, you'll get a falsely low result.
So how do they protect them?
These samples are often collected in special amber-colored tubes that block light, or the standard tubes immediately wrapped in aluminum foil or placed in a light-blocking bag right after collection.
That's a lot of variables to manage. It seems like there's so many ways things could go slightly wrong. So practically speaking, how do labs and phlebotomists actually prevent these issues day to day in the real world?
Well, our sources really emphasize several key practical prevention strategies. For example, to prevent hemolysis, those bursting red blood cells we keep mentioning, it's critical to use gentle inversion, maybe 5 to 10 times depending on the tube, not vigorous shaking, when mixing tubes with additives.
Gentle inversion, not shaking. Got it.
They also need to use an appropriate needle size. Too small a needle can shear the cells as they pass through. Avoiding prolonged tourniquet application more than a minute is important. Ensuring the venipuncture site is completely dry after cleansing with alcohol, because residual alcohol can hemolyze cells. And as we mentioned for capillary sticks, don't excessively milk the site.
Okay.
And perhaps the most critical step of all, absolutely non-negotiable, is proper patient identification.
Right. Making sure the sample belongs to the right person.
Exactly. This means using at least 2 unique patient identifiers, typically the patient's full name and their date of birth or medical record number, and verifying these identifiers actively at the bedside or draw station by comparing the requisition form information to what the patient states, and crucially to the information on the patient's identification wristband, if they have one.
And the labeling.
The specimen tubes must be labeled immediately after collection, right there, in the patient's presence. Never label tubes beforehand, and never take unlabeled tubes away from the patient. It's about diligence. Every single step, creating multiple barriers against error.
It really does sound like a remarkable ballet of precision and vigilance, doesn't it? Where every single step is interconnected and absolutely crucial for the final result. But let's zoom out just a bit now. Beyond all these technicalities, what are the broader implications of this entire process for patient safety, for public trust, and, well, for ethical practice in healthcare?
That's a great question, and it brings us to a really crucial point. The human element woven through all of this technology and procedure. What truly stands out in the material is how these layers of ethical and safety considerations underpin every single step.
Like consent.
Exactly. Patient consent isn't just, good practice. It's a fundamental ethical and legal requirement. It upholds patient autonomy, Their right to understand what procedure is being done, why it's being done, the risks and benefits, and importantly, their right to agree to it or to refuse it.
And that's for every blood draw.
Generally, implied consent covers routine, low-risk procedures, like venipuncture once a patient presents for the test. But for higher-risk procedures, think amniocentesis, invasive biopsies, or for tests with significant legal or social implications, like genetic testing or forensic analysis, specific written informed consent is often required to clearly document that detailed understanding and agreement.
Okay. What about privacy?
Patient privacy and confidentiality are paramount. This is rigorously governed by regulations like HIPAA here in the U.S., which protects all individually identifiable health information, PHI. Labs have strict policies to ensure patient data is secure.
And for legal samples?
Right. For specimens that might be used in legal proceedings like forensics or workplace drug testing, there's also a rigorous chain of custody protocol. This meticulously documents every single person who handles the specimen, from collection to testing, to ensure its integrity and prevent any possibility of tampering or mix-up.
That makes sense. And safety for the healthcare worker, too.
Absolutely essential.
Okay.
Biosafety precautions are mandatory. This starts with universal precautions, the principle of treating all blood and bodily fluids as potentially infectious, regardless of the patient's known status. Then there are engineering controls, like using puncture-resistant sharps containers for needle disposal or safety-engineered devices like self-sheathing needles. Work practice controls are also key things, like meticulous hand washing before and after patient contact, and absolutely no eating, drinking, or applying cosmetics in lab work areas. And finally, mandatory personal protective equipment, or PPE gloves always, lab coats or gowns, and eye protection like goggles or face shields when there's any risk of splashes. It's all about protecting both the patient and the staff.
That level of professionalism and strict adherence to protocols seems truly foundational, doesn't it? It connects directly back to accuracy and building that essential trust.
It absolutely does. Professionalism in this context encompasses competency, ensuring staff are properly trained and their skills regularly assessed. It means thorough ongoing training on procedures and safety. It demands strict adherence to those standard operating procedures we discussed. And it requires meticulous, unwavering attention to patient identification and specimen labeling. All of these elements work together to prevent errors, minimize risks, prevent potential malpractice, and ultimately ensure reliable results. And reliable results are what build confidence and trust, the patient's trust in their diagnosis, the clinician's trust in the lab data, and the public's trust in the healthcare system. It's really the silent promise of quality in healthcare.
That trust is so vital, which leads me to the final question, really. How do laboratories maintain and continuously improve quality in such a complex, high-stakes environment? It sounds like an endless battle against potential variables and errors.
It is, in many ways, an ongoing battle. But labs achieve remarkably high levels of quality through robust, multi-layered systems. It's almost like building a multi-layered defense shield against error.
How does that shield work?
Firstly, there's rigorous accreditation. Labs are typically accredited by external independent bodies like CLIA, Clinical Laboratory Improvement Amendments in the U.S., ISO 15189 internationally, or CAP, College of American Pathologists. These bodies set extremely stringent standards for everything from personnel qualifications to procedures to quality control, and labs have to undergo regular inspections to maintain their accreditation.
So external oversight, what about internally?
Internally, they constantly monitor their own work through something called internal quality control, or IQC. This involves running control samples with known values alongside patient samples every day, or even multiple times a day for some tests. They use statistical tools like Levy-Jennings charts and complex Westgard rules to analyze these control results and detect even subtle shifts or trends that might indicate a problem with the test system, like reagent degradation or instrument malfunction, before it affects patient results. Calibration verification is also part of this.
Okay, so constant self-checking.
Yes. And crucially, there's a deep commitment to continuous quality improvement, or CQI. This isn't just about fixing problems. It's about proactively making processes better. Labs often apply methodologies borrowed from industry, like Lean, to streamline workflows, eliminate waste, and improve efficiency. Or Six Sigma, which aims to drastically reduce variations and defects in processes to achieve near-perfect results.
Lean and Six Sigma in the lab. Interesting.
Very much so. They also have clever monitoring systems. One is called Delta Checks. This is where the lab's computer system automatically compares a patient's current test result to their previous results for the same test, if available.
Ah, looking for big jumps.
Exactly. If there's a significant, unexpected difference, it flags it for review. It could be a real change in the patient's condition, of course, but it could also signal a potential pre-analytical error, like a mislabeled sample or an interfering substance. It's another safety net. And of course, there's a critical system for reporting critical values, results that are potentially life-threatening directly and immediately to the responsible clinician, bypassing standard reporting channels to ensure rapid intervention.
So it really is a never-ending cycle of checking, analyzing, improving, and safeguarding.
It has to be. The stakes are simply too high. It's a constant cycle of self-assessment and striving for perfection.
Wow. Well, we've certainly journeyed through the incredibly intricate world of specimen collection today, realizing, I think, that the smallest details, the angle of the needle, the temperature of the tube, the order of draw, they truly have the biggest impact on diagnosis and ultimately on your care as a patient. It's truly a hidden world of vigilance, precision, and just continuous dedication to getting it absolutely right.
Indeed. The reliability of every single diagnostic step that follows, every treatment decision that's made based on those results, it really does begin with the quality of that tiny, carefully collected sample. It's honestly a profound testament to the vigilance, the training, and the expertise of countless phlebotomists, nurses, medical assistants, and lab professionals that so many critical medical decisions can reliably begin with these often unseen foundational steps.
So, the next time you or perhaps a loved one has a lab test, maybe take a moment to consider the unseen heroics behind that accurate result you receive. And maybe ask yourself, what aspect of this deep dive made you rethink the diagnostic process the most? Keep asking those questions, keep being curious, and keep diving deep.